Assalamualaikum. I am IRDINA SYAZA BINTI SAHBUDIN. Currently, I am doing my final year project on a topic, the removal of manganese ions from water treatment effluent by using rice husk. My supervisor is IR Nabil Abdul Azim bin Saifuddin and my co-supervisor is Encik Abdul Rahim bin Abdul Aziz, a chemist in the Sungai Baru water treatment plant. The main objective of this study is to solve the problem faced by Sungai Baru water treatment plant. The problem is the high concentration of heavy metals in the effluent from Sungai Baru water treatment plant. Based on the previous studies, there are various methods have developed to solve the problems. This study will focus on the most efficient method in solving the problem, which is bioabsorption. Three parameters such as pH, contact time and adsorbent dosage will be observed throughout this study. To start the studies, I have found that there are various types of biomass used in solving this problem. This presentation will explain the ability of each of the biomass to absorb the heavy metals. In order to achieve the objective, I have chosen the best biomass, which is rice husk. There are two ways in preparing rice husk that I will present. Three isotopes are involved in this study to study the relation of the heavy metals and the bioabsorbent. This is the outline for my presentation. I will present the introduction, literature review, methodology, and expected result of my studies. The introduction part will include problem statement, research question and research objective, and scope of studies. In literature review, I will present on the absorption, manganese, effluent, and rice husk. For the next chapter, I will present on the methodology, which include research flowchart, batch absorption studies, and absorption isotope study. Lastly, we will go through the expected result and reference. The first part of this presentation is introduction. The problem statement of this study is the concentration of manganese in the effluent. The recorded concentration is nearly reached to the maximum limit. The high concentration of manganese affects living organisms in the water and it expands through the food chain and could influence human health and living organisms. Thus, the effluents that contain high concentration cannot be released to the river. Next is the development of various methods for removing heavy metals give different ability. And lastly, the abundance of raw materials. The availability of rice husk in large quantity can affect the environment. Farmers usually burn the rice husk and it will release carbon dioxide, which is harmful to humans. In this study, there are three research questions that need to justify at the end of this research. The first question is how to prepare and characterize the adsorbent made from rice husk. Question 2 is how do pH, bioadsorbent concentration and contact time affect the percentage removal of manganese from the effluent? And last question is what is the relation isotope of manganese removal with rice husk as the bioabsorbent? The research objective is to prepare and characterize the adsorbent from rice husk. Second is to perform the parametric study of manganese removal from the effluent. Lastly is to study the absorption isotope for the process. All the objective is to justify and answer the research question. This part I will present the scope of this study. The purpose of this study is to treat effluent produced by Sungai Baru water treatment plant. This plant is operated by Syarikat Air Darul Aman, located in Sungai Baru Simpang 4, Perlis. The sample taken is the effluent from water treatment plant. For this study, the samples are collected at the final discharge point at the lagoon. To collect new and fresh effluent, it will be collected after tank cleaning process. This study only focus on three parameters, which are pH, adsorbent dosage, and contact time. The main equipment that involved in this study are FTIR, SEM, DR6000, and furnace. All this equipment is to characterize the rice husk and to determine the concentration of manganese. As I am doing the study under Syarikat Air Darul Aman, I will follow the protocol and ethical management of the company. Second chapter of this study is the literature review. In this chapter, I will also present the water treatment process involved in the plant. Water treatment process in this plant is a process to treat raw water from Arau Canal before being delivered to Pulau Langkawi. The raw water from Arau Canal undergo 6 primary processes with high technology and specialist workforce who manage the water treatment plant. The process starts at raw water intake, where there are pumps that collect water from the river. Then the raw water inlet chamber and aerator to supply oxygen. At this process, aerator also convert iron and manganese element into insoluble. The mixing chamber after the aerator is to mix the alum with raw water. Flocculation tank enhance the interaction between particles, forming new aggregates that can be separated more easily. In the sedimentation tank, the suspended solid will settle at the bottom of the tank. Filtration tank function to trap small particles residue that did not settle in sedimentation tank. The last process is post-chemical dosing. There are 3 types of chemicals that will be dosed at this process. Lastly, the clean and safe water being stored in treated water tank before being sent to Pulau Langkawi. What is effluent? In this study, effluent refers to the wastewater from water treatment plant. The effluent is produced from tank cleaning process. The cleaning process of flocculation and sedimentation tank happen once a month. Filter tank undergo filter backwashing every 72 hours. The water from the cleaning process will flow to the slush tank and released to the lagoon. The effluent cannot be discharged to the river if contain high concentration of manganese. According to the Environmental Quality Act, the acceptable concentration of manganese is 1.0 mg per liter. Currently, concentration of manganese in the effluent is 0.66 mg per liter. The value is near to the accepted limit. Hence, this study is to prevent the concentration exceed the standard. Manganese is a natural element found in rock, soil, and water. It is essential nutrient for microorganisms, plants, and animals. Manganese compound exists as solid in the soil, while as solids and small particles in water. Commonly, the source of environment exposure to the manganese in the effluent is from anthropogenic sources. Anthropogenic sources include municipal wastewater discharge and sewage sludge. Manganese is often transported from the source to the river by suspended sediments. The manganese is absorbed to the suspended sediments. As the manganese is soluble compound, there is tendency of the soil to absorb the manganese. The interdependent is a process where manganese exchange between water and sediments. This process changes the manganese from sediments to water. This is how manganese occurs in water. In this slide, I will present the various methods used in separating heavy metals from effluent. The methods are oxidation, ion exchange, electrochemical, coagulation and flocculation, ozonation, membrane filtration, photochemical, and irradiation. All these methods have been developed from the previous studies. All these methods have the advantages and disadvantages. From all these methods, adsorption and ion exchange are the most recognized methods. However, ion exchange need high investment and regeneration costs. Hence, this study will focus on the adsorption method. So, what is adsorption method? Adsorption is a separation of a substance from the surface to another surface. There are 2 types of adsorption, which are chemical and physical adsorption. Physical process is a metal's ion are adsorbed electrostatically across the surface of the materials, and chemical process is a chemical bonding between adsorbate and the surface of adsorbent. To test the type of adsorption involved in this study, I used DR isotherm. Biosorbent is the adsorption process that uses natural biomaterials as raw materials. There are many types of biomaterials that can be used as bioadsorbent. The table shows the comparison between 4 types of bioadsorbent. The data is taken from the previous studies. According to the Ardicola et al., rice husk has the ability to remove 100% of manganese within 100 minutes. From the comparison, I have chosen rice husk as the raw materials in this study. What is rice husk? Is it the thing we eat every day? No. Rice husk is the hard protein that covered the rice. There are abundance of rice husk in Malaysia. Malaysia produces 408,000 million tons of rice husk per year. Rice husk is the waste from rice process. The way farmers manage rice husk is by burning. The burning of rice husks are not safe to the environment. The burning will produce carbon dioxide that is harmful to humans. Based on the previous study, rice husk is effective adsorbent. The last part for this proposal presentation is methodology. This slide presents the research flow process. The study starts with collecting rice husk from rice factory in Perlis. I will collect 3 kg of rice husk. Then I will prepare the rice husk for the adsorption process. The rice husk is then being characterized. The next process is to do the batch adsorption study and adsorption isotherm study to fulfill the objective of this research. How to prepare the rice husk? There are 2 methods in preparing rice husk, which are with pre-treatment and without pre-treatment. Pre-treatment method start with milling the rice husk, then wash with distilled water to remove the impurities particles that attach to the rice husk. After washing process, oven dry the rice husk at 105 degrees Celsius for 5 to 6 hours. After drying, sieve the rice husk with 250 micrometer particle size. After sieving process, purify the small particles with HNO3 for overnight. After a night, wash them again to neutralize the rice husk, then etch them using furnace at 550 degrees Celsius. The second method in preparing the rice husk is without pre-treatment. After collecting the rice husk, oven dry them at 60 degrees Celsius for 3 hours. Then dry them again with a dryer before being stored in dry place. Characterization of rice husk is done by using FTIR and SEM. In this slide, I will present the method for parametric study. The first process is to do the baseline. This process is to dilute effluent with distilled water to remove suspended solid. After the dilution, test the concentration of manganese using DR6000. Next is test the effect of pH. Set up effluent with different pH value from 1 to 8. Then mix 20 ml of effluent with 0.1 gram of rice husk ash. Shake the mixture using shaker for 5 hours at 130 rpm. To test the effect of contact time, set up effluent with constant pH, then mix 20 ml of the effluent with 0.1 gram of rice husk ash. Shake the mixture using shaker. Take out the sample one by one for every 60 minutes. To test for adsorbent dosage, set up effluent with constant pH value at pH 3, then mix 20 ml of effluent with different adsorbent dosage starting from 0.1 gram to 0.8 gram. Shake the mixture using shaker for 5 hours at 130 rpm. The last process for the parameters test is to test the final concentration of manganese. After takeout from the shaker, filter the mixture using Whitman filter paper, then determine the concentration reading using DR6000. In this study, there are 3 adsorption isotopes involved to test how the adsorbent interact with manganese. The 3 adsorption isotopes are Langmuir isotope, Freundlich isotope, and DR isotope. Langmuir isotope relate the capacity of adsorption and adsorption rate. Freundlich isotope is to study the relationship between the solvent adsorption and the adsorbent surface. DR isotope is used to identify either the type adsorption is physical or chemical adsorption. The last part for my presentation is the expected result of this study. The result to achieve Objective 1 can be seen through FTIR and SEM. This figure shows the result of rice husk ash from SEM. The expected result for Objective 2 are the optimum pH for manganese removal is at pH 6, contact time 60 minutes, and adsorbent dosage is 0.5 gram. Lastly, the expected result for Objective 3 is the type of adsorption is physical sorption, which have E value lower than 8 kJ per mole. The adsorption intensity value is low to get a strong interaction between manganese and rice husk ash surface. The next slide is the reference I used throughout this study. That's all for my proposal presentation. I hope my proposal presentation is clear. Thank you.